Hello, and welcome to Biology 111, Anatomy and Physiology, Part 1. This is the first half of a full-year course in anatomy and physiology that's aimed at college nursing students and other students going into the health sciences. Normally, this would be taught in a classroom. Remember those? Due to the current COVID-19 situation, myself, like many other instructors, have had to move to an online format. I spend much of my time sitting in a darkened room, talking to myself and slowly going mad. Please join me on my journey. Now, I should point out that I'm new to this, and it starts out a bit rough, but the sound quality and the presentation quality get better, so please stick with it. I figured that because I have to record this, I might as well put it on the internet in the hopes that someone else will find it helpful or at least interesting. If you have any questions or comments, please feel free to leave them below. Please try to be nice, but if you can't, at least try to be clever. Although the quality of the PowerPoints will improve, the quality of my humor will not. Anatomy and Physiology Part 1 and 2 covers a lot of material, and I like to be sure that my students are all on the same footing when we get started into the tougher stuff. For that reason, you might find that the first third or so of Anatomy and Physiology Part 1 seems almost remedial. It's stuff that you should have learned in high school, but maybe you slept through it. If you didn't sleep through it, bravo, you might want to skip ahead a bit. Anyway, let's get going. So let's start with the absolute basics here. We're talking about anatomy and physiology, and we should probably define those two terms. Now, if I had a class in front of me, I would have a bit of a discussion with you guys and talk about, what is anatomy, what is physiology, can you give me examples of those two things? But unfortunately, I'm going to have to kind of answer my own questions here. I got to say, it's really quite weird sitting in a dark room and talking to yourself while you're doing this. But, well, that's what we got to do this term, unfortunately. Okay, so what is anatomy? Now, anatomy is the study of structure, basically. So we're looking at structures within the body. We might be looking at muscles and how they're arranged. We might be looking at structures in a cell, talking about the compartments in a cell, vacuoles and mitochondria and the nucleus and all that sort of stuff. So we're looking at physical structures, components of the body, components of cells, etc. Physiology relates to function. So now we're looking at the function of those structures. And I'm always going to try to tie the two together. So for instance, the lungs, they contain alveoli, little sacs that you breathe the air into. And the function of those little sacs is to increase surface area. And the reason for that is that we have a huge surface area. We can exchange gases, carbon dioxide and oxygen, between the air and the blood very quickly. The larger the surface area, the more quickly we can exchange stuff. We can get oxygen in faster, we can get carbon dioxide out quicker. So the anatomy, the fact that the lungs are made up of lots of tiny, tiny little sacs, instead of just being like big bags, relates to the physiology. The anatomy has a distinct function to it. And there's an expression in biology that form fits function. So form, anatomy or structure, fits the function, the physiology. So we have evolved to have structures that work well. They do their job well. And this surface area problem that I just talked about is something we'll come back to quite often. In the kidney, for instance, we need to have rapid exchanges between the blood that's coming into the kidney and the urine that's leaving. So within the kidney, you're extracting waste and you're reabsorbing things that you might want to hold on to. And we'll come back to that later. But generally, the anatomy of a structure tells you a lot about what it does. and vice versa. So in this class, what we're trying to do is we're trying to look at the anatomy, and you're going to have to learn the anatomy. And unfortunately, in this class, there's lots of jargon and terminology that you are going to have to learn. And that's because we have so many bits and pieces, and they all have to have names so we can talk about them. So there is a fair bit of memorization when it comes to the anatomy component. But always think about the anatomy as it relates to the physiology. So why is this certain structure shaped the way it is? Why does it have certain properties? It's related to its function. And I'm always going to try to relate form and function, or anatomy and physiology, as we go through the course. We are, of course, living things. Fundamentally speaking, all the things that our cells need to accomplish and that our bodies need to accomplish are the same things that every other living thing needs to accomplish. So I thought it would be useful first to define life and figure out what living things need. What do they have to do to be living things? I'm not entirely sure that life cereal is still a thing. Is it? Did this joke not land? I'm not sure. Anyway, I'm sticking by it. Living things come in lots of different forms. So next to the life cereal ad there, we've got some bacteria, some spirochete bacteria, which we'll talk about in Bio 105, which you might be doing with me next term. We've got a plant. Plant needs to do all the same things we need to do. Plants are kind of neat in that they can also make their own food using sunlight, something we can't do. Next to that, we've got some yeast cells, which, they might seem rather simple, but those cells have to do the same stuff that all of our cells have to do. On the bottom and the left there, we've got something called Giardia, which is an infectious protist. It's a single-celled eukaryote, and don't worry too much about all those terms just yet, but this is something that causes explosive diarrhea. It causes something known as beaver fever, which you might have heard of. But again, this cell has to do all the same things that all of our cells have to do. Fungus, Acheta, doesn't matter. All of the cells that make up these organisms have to do some very specific things. Now again, if I had you in front of me, we would sit down and talk about what these things are. But I've come up with a pretty good succinct list for you. Now you might think that biologists have a pretty good handle on what life is. Not necessarily. If we're defining life, we can't look at one single characteristic. So you might think reproduction would be a good characteristic of living things. But there are some things out there that reproduce that aren't generally considered to be alive. A computer virus would be a good example. So computer virus will use your computer to make copies of itself, but I think most people would agree that a computer virus is not technically alive, at least not in a biological sense. And even a biological virus, like a coronavirus, for instance, is that alive? It can reproduce, but it doesn't have its own metabolism. It can't sustain itself on its own. It has to infect other cells, just like a computer virus has to infect computers. So when it comes to defining life, most biologists would agree there is no single defining feature. It's a set of features. And I've come up with a few that I think are applicable to all living things. Now there's some things that aren't here, and if I were to ask this question on a test, hint maybe, you don't have to give the ones I've got here, the 7 I've got listed here. You might have some good ones yourself, and I invite you to offer those up as well. But I think when it comes to defining life, living things need to have these characteristics, or at least most of these characteristics. So living things possess order. Now what do I mean by that? Well, living things are complicated, even the simplest of living things. Simple bacteria, they do a lot of remarkable stuff, and they have ordered molecules within them. They have DNA, they have proteins, they have lipids. These are highly ordered molecules. And they have to maintain that order. Now we'll talk about this later, but you may have heard of the word entropy before. And entropy refers to a tendency towards disorder. So in the universe, things tend towards randomness. If you have a complex molecule and you just let it sit out in the environment, it's going to break down. If, let's say, you take a drop of dye and you add it to a bathtub full of water, the molecules bump into each other, they rearrange themselves, they move all over the place, and they randomize themselves. They basically will randomly distribute themselves throughout the bath water. So unless something is applying energy to maintain structure and maintain order, order breaks down. Personally, I think maintaining order is one of the key aspects of life. Living cells and living things fight disorder. They expend energy in order to maintain order. And really, the food you eat, the oxygen you breathe in, your cells in your body are using that oxygen to liberate energy from your food, and that energy is being used to maintain the order of your cells. It's being used to maintain the order of your DNA and your proteins and so on. Once you stop doing that, the order breaks down. You spend your entire life fighting entropy. You spend your entire life using energy to fight breakdown, to fight disorder. And of course, when you die, you lose that battle and your body breaks down. Reproduction is another key aspect of living things. But as we talked about, there are some non-living things that can reproduce. So living things, living cells, living organisms make copies of themselves. Otherwise, they go out of existence, they go extinct. Growth. So even if we're talking about a single cell, a single-celled organism like an amoeba or a paramecium or something like that, they grow over their lifespan. So with an amoeba, it divides into two little amoeba, and they grow into bigger amoeba, and then they divide into two little amoeba, etc. And of course, if we're talking about complex organisms like humans, we start out as a single cell, and we grow into something that consists of a few 100 trillion cells, which is really remarkable when you think about it. So your parents contributed a sperm and an egg. Maybe don't think about that part, but the sperm and the egg came together and they produced a cell called a zygote, and then that cell, over many, 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 many divisions, grew into you. Energy utilization, and typically this is all summed up as metabolism. So metabolism is the set of chemical reactions that occur in every cell in your body. And what metabolism does is it makes energy available to you for growth and reproduction and maintaining order and all the other stuff you have to do. Next, living cells and living organisms need to respond to the environment. So if we look at a single-celled organism like an amoeba, if the environment becomes, let's say, more acidic or more basic, the cell has to respond to that. Otherwise, it's not going to survive. And the same thing happens in your body. Cells have to respond to changes in the internal environment, the environment within your body. And that relates to homeostasis. Homeo means same. We're talking about maintaining the same environment within your body and within your cells. What I find quite fascinating is that the first cells evolved about 3.5, 3.8 billion years ago, and they maintain the environment that they evolved in. So the environment within a cell is very similar in terms of salinity and pH to the very ancient oceans where those cells first evolved. every cell has to maintain its pH, it has to maintain other properties inside that cell. Your body maintains a specific temperature, etc., so that all of those cells are happy. So if the environment changes, the cell will respond to the environment and maintain its internal environment. The internal environment of the cell stays about the same. And finally, evolutionary adaptation. So cells and organisms do change over time. And this isn't a willful thing. It's just due to the fact that cells and organisms that aren't adapted to their environment die out. The ones that are adapted to their environment live on, reproduce, and pass the genetic material that determines their specific properties to the next generation. So life will evolve and change over time to match the environment and to match the needs of that organism. So as I mentioned, I think these are a pretty good list of factors that define life. You could probably come up with a few more. You might say things like getting rid of waste, taking in food, that kind of thing. I would say that those are part of energy utilization. Energy utilization doesn't have to be taking in food and breaking it down. It could also be making your own food using photons of light, which is what plants do. But we'll come back to metabolism later, and we'll talk about it in lots of detail, maybe even more detail than you want to know. While we're looking at the big picture, we should look at biological hierarchies. So a hierarchy is where you have a category that's part of a bigger category, that's part of a bigger category, etc. And hierarchies exist in living things and exist in the universe in general. I mean, look at our own solar system. We have a star, the sun, that's being orbited by planets. The distances between the planets are immense, but we have this unit we call the solar system. And stars and solar systems make up a much bigger unit that we refer to as galaxies. And galaxies make up the universe. But we can go the other way and look at really small stuff. So atoms are made up of little bits. They're made up of protons and neutrons that form the nucleus and electrons that orbit around the nucleus. And most of the atom is empty space, but we have these parts. Atoms come together and they form molecules like amino acids. And amino acids and other molecules come together and form big molecules called macromolecules. So amino acids form proteins, for instance. And then those macromolecules, those big molecules, form organelles. They form things like membranes and so on that form organelles. And the organelles are little compartments within a cell. So the organelles group together and they form a cell. And cells, when you put them all together, they form tissues. Tissues are collections of cells that are working together to perform a certain function. So smooth muscle cells, they are responsible for contractions that create peristalsis, for instance, in your esophagus and your intestines. These are contractions that you don't have any control over. And all those smooth muscle cells are working together. There are some tissues, like the tissues that make up the small intestine, for instance, that consist of different cells, different cell types, but they're all working together. Tissues form organs, and an organ is performing one particular function. So your intestine is involved in absorbing nutrition. Your blood vessels are involved in moving blood throughout your body. The organs are part of an organ system. So a blood vessel is part of your circulatory system or cardiovascular system. The intestine is part of your digestive system. And then finally, all those systems come together and they form an organism. They form a complete living being, like yourself. To examine this hierarchy of structure, we have a hierarchy of biological fields. So if we're looking at the entire organism, let's say we're looking at an entire human, for instance, we're talking about gross anatomy. So this is looking at structures that you can see with your naked eye. So if you're doing a dissection, let's say we're dissecting fetal pigs, or you're dissecting cats, or you're dissecting a human cadaver, this is gross anatomy. Not gross because it's kind of gross to do, but gross meaning big scale, essentially. So we're looking at organ systems and organs. So if you dissect a fetal pig in the lab, you might identify the digestive system, which would be an organ system, and then look at the individual organs. Look at the stomach, look at the small intestine, the large intestine, and look at the structure of those organs. But it's all stuff that you can see with your naked eye. And we usually say that it's macroscopic, macro meaning big. You can see it without a microscope. Now perhaps you're going to take a sample of preserved tissue from one of those dissected specimens, slice it really, really thin, look at that on a slide under the microscope. That would be histology. So histology, we're looking at tissues. So let's say we took a section of the small intestine, and we're going to look at the different layers, the smooth muscle, the epithelium, etc. within the outer wall of the intestine. We would be looking at histology. Now we could zoom in further and look at a single cell. And the study of cells is called cytology. Cyto means cell. So if you see cyto in a word, it refers to a cell. So in cytology, we're looking at entire cells, and we're also looking at the parts of cells. We might be looking at organelles, vacuoles, the nucleus. Those show up under a light microscope. If you want to look at smaller organelles, like for instance the mitochondria, ribosomes, you need an electron microscope to do that. The next step down, so again we're looking at smaller and smaller bits, would be molecular biology and biochemistry. Now here we're looking at the role played by macromolecules. We're looking at things like DNA and proteins and so on. Can't actually see these things not even under a microscope, except for really, really high-powered electron microscopes. But generally we're looking at reactions. We're isolating out components like DNA and protein and so on, but we're looking at smaller bits. Then beyond that, organic chemistry, we're looking at the components of these macromolecules. Typically, we're looking at amino acids, for instance. We're looking at nucleotides, that kind of stuff. And chemistry, now chemistry can involve a lot of stuff. It could involve some of the larger molecules, but in this example, I'm talking about chemical reactions between atoms. And of course, that's something that you can study in a test tube. You're not actually looking at the atoms, but you're studying the reactions that occur. Again, with gross or macroscopic anatomy, we're looking at large structures. We're looking at the entire organism, an entire human. In this case, this is a dissected cadaver here. You can see the neck at the top and the chest down the bottom. We're looking at muscular systems, we're looking at digestive systems, we're looking at nervous system, etc. But we're looking at big things that you can see with your naked eye. What you're seeing here is a dissected cat. We used to do cat dissections in the lab in CNC. We don't so much anymore, although I may have a few demo dissections. It's difficult to get the cats anymore. I used to like to use cats because these are cats that come from Mexico. They were rounded up because they were feral cats that were a problem, so they were being euthanized, and we were just getting them as a byproduct of that. But within this cat here, we're seeing the same structures, of course. We're seeing the heart very clearly there. We're seeing the liver. Heart's part of the circulatory system. Liver is an accessory organ of the digestive system, as we'll talk about. But you're looking at things that are very large, that are macroscopic. You can see them with your naked eye. So let's just go through, before I look at the other levels, let's just go through the gross anatomy that we will be focused on in Bio 111 this term. The first organ that we're going to look at is the integumentary system. So the integument is a fancy name for the skin. The skin is technically an organ. It's the largest organ of your body, followed by the liver, which is the largest internal organ. But the integumentary system is the outer layer of your body. There's an epithelial layer that makes contact with the external environment, of course, and protects you. And then underneath that, we have a connective layer known as the dermis. And we'll get into all of that later on. The skeletal system, my personal favorite. I find the development of the skeletal system quite fascinating. The structure of the skeletal system, I think, is quite fascinating as well. We'll spend quite a bit of time on this in the lab because there is a lot of terminology to learn, but I think you'll be quite satisfied when you learn those terms, when you're able to identify the bones and identify the little bumps on the bones and things like that, and know what they do. Every single bump on that skeleton is an attachment point for a muscle. The little holes are where certain arteries and veins exit, etc. The skeleton, every bit serves some sort of function. The next system we'll look at is the endocrine system, and this consists of a large collection of glands distributed throughout the body that produce hormones, which are signaling molecules that are released into the bloodstream. Now, if you have done 111 with me before, you may be wondering why we're doing this year, because normally I have taught this as part of Bio 112. So normally I've taught the endocrine system in second term, right after we look at blood and circulation. I think that kind of makes sense. But it's being traditional to teach this as part of 111 in Prince George. And because I am teaching a lot of Prince George students this year, I thought it would make more sense to move this from 112 to 111 so that things mesh a bit better. And the last macroscopic system that we'll look at in Bio 111 is the nervous system, and we'll spend quite a bit of time on this. So the brain and the spinal cord together form the central nervous system, and then we have nerves that radiate off of that make up the peripheral nervous system. After you understand the fundamentals, we will get into the remainder of the macroscopic systems next term in Bio 112. So we're going to look at the muscular system, the cardiovascular system, so that's the heart, veins, and arteries, the lymphatic system, that'll also be covered in Bio 105. So Bio 105 is microbiology, and we'll get more into the immune system, which is very, very closely linked with the lymphatic system. We'll talk about the respiratory system, we'll talk about the digestive system, urinary system, and the reproductive system. So stay tuned for that. So how might we go about studying gross anatomy? Well, if the specimen is no longer living, that's not a big issue. We can do a dissection or post-mortem. But what if it's a patient and we want to see what's going on inside that patient? We could do exploratory surgery, but that probably wouldn't be the first thing to do. There might be other means we could use to peer inside without actually having to cut the patient open and risk damage and infection. X-rays are quite good for viewing dense structures within a body, within a human. The thing is that the structures have to be dense, though. They have to be dense enough to absorb X-rays. So what you do in radiography, which is the use of X-rays to look at internal structures, what you do is you shine X-rays through a patient. X-rays are a form of electromagnetic radiation. Light, the light that we see, is also a form of electromagnetic radiation, but of course we can't see X-rays. So we have a source of X-rays, the X-rays pass through the patient, and then we have a piece of photographic film on the other side of the patient, or a sensor of some sort, which would be the way of doing it nowadays. Using film is a bit old school. But let's look at the example of film because maybe it's a bit easier to understand. So imagine we had a piece of film behind the patient. If the X-rays get through, they will expose the film and they will turn it black. If the x-rays don't get through because they're blocked by something dense, they will not expose the film and the film will remain white. So what you're seeing here on the left is a radiograph, so an x-ray photograph of a person's upper arm. So we're seeing the humerus there and it's broken. And the white that we're seeing, the bone, the humerus, is much denser than the surrounding tissue, so it is blocking x-rays. The x-rays are not getting to the film and they're not exposing it. Again, though, if you used a sensor and you used digital equipment and a computer monitor, which is the way it's pretty much always done nowadays, you get the same result. On the right here, you're seeing a mammogram. So we're looking at a breast, and again, we're passing x-rays through this tissue, and there's a tumor there. Tumors are comprised of lots of cells that are clumped very closely together. So this tumor consists of rather small, dense cells all packed together, and that white spot is where the x-rays have been absorbed because of that density. Now, one side effect of x-rays is because x-rays are kind of high energy beams of radiation, they can potentially damage DNA within cells, and that can cause mutations within the cells, which could perhaps lead to a cancerous cell, something we'll talk about more. Now, the damage from x-rays is really slight. X-rays have gotten much, much better. We've got more sensitive sensors now that can detect x-rays and blocked x-rays. So it's not nearly as problematic as it was in the past. But when you use x-rays, there is a slight concern that you may induce mutations. Again, it's not generally a big problem unless you're using high doses of x-rays and you're doing it often. I've got a couple real-world examples of x-rays for you to look at. The first one here is actually of my stepdaughter, and this is when she was about 8 years old. She was holding a leash, and at the other end of the leash was a very hyperactive collie that decided to take off and pull her abruptly, and she fell to the ground with her arm outstretched. And you can see that we've got a broken collarbone or clavicle. And it's a pretty significant break. You can see the bone is displaced quite a bit. Now, that's not a huge issue in a young child necessarily. What has to be done is that the bones have to be reset, so they need to be put back together and aligned properly. But fortunately, children are quite resilient. And we'll talk about this when we talk about bone development, and the bones do tend to mend very well, and they mended quite well in her. So again, what you're seeing here is we've got x-rays that were passed through her. There's a sensor on the other side of her body, and the sensor will pick up the x-rays that get through areas where there's not much to inhibit the x-rays. Like, for instance, the chest cavity where the lungs are will appear quite dark. Areas where there is very dense material like bone that will block the x-rays will appear white. Now, the other thing that's kind of interesting here that we'll get back to when we talk about the development of the skeleton, but you might be, interested in at the moment, we can see that we've got this line along here. This is the humerus. We've got the top of the humerus up here and the bottom of the humerus down here. And you might be wondering why they're not attached. Well, that's because she's still growing, and that division between the upper portion, the head of the humerus, and the bottom shaft of the humerus is where growth occurs. So between that line, new bone is being produced, and that allows the humerus to elongate because, of course, as you grow, your bones are going to elongate. But we'll come back to that, and I find that stuff really quite fascinating. Now, skip forward another year, and well, I hate to say it, but she's a bit accident prone, and she twisted her ankle rather badly and had another set of x-rays taken of her foot. As it turned out, there was no breakage, so this is a fairly normal x-ray here. And you'll have to excuse my really bad pun there at the top. That's about as good as my humor gets. I apologize. Anyway, once again, the other thing that's kind of interesting here is we've got these growth plates here. So you can see that the heads of the bones are not attached to the shaft. So we have the bones here not being connected because the long bones still have to grow. So that's totally normal. I mean, if you didn't know that, you might look at this and think that the bones are broken. But of course, you won't think that after we get through the skeletal portion of this course. Pay attention to the terms at the bottom. We'll come back to that. But we've got a superior view, which is looking down on the foot. We've got a posterior view, which is looking at the back of the foot, looking at the heel, which is called the calcaneus bone. We'll come back to that, but that's what you're seeing right here. I'll just put an H for heel for now. And then we've got a lateral view. Lateral means from the side. So this is the heel or calcaneus bone. And you can see that even it has its own growth plate. So the heel needs to expand. This piece here needs to be free so it can grow out, and new bone can be deposited at that growth plate or epiphyseal line. We'll talk about that later. Radiography, the use of X-rays, can also be used to look for abnormalities or blockages in blood vessels and other tubes, like for instance the ureters that conduct urine. So we talked about how we can use X-rays with soft tissue before, and we talked about looking for tumors in the breast, for instance. What you're seeing here on the left is an angiogram. Angio means vessel. So angio in Latin just means vessel. We're looking at blood vessels here. And what's being done is they've taken what's called a radiocontrast agent. And this is a fluid that's quite dense. And they've pumped that into some blood vessels. That circled area should contain some of that radiocontrast agent, but it doesn't. So that blood vessel continues in that space, but the agent couldn't get in there because there's some sort of blockage. So an angiograph is going to allow us to see blood vessels by using this radiocontrast agent, and also identify areas that are blocked. And if an area is blocked, of course, it won't conduct blood, so that's a problem. The urogram here, we're looking at the kidney. And they have injected a radiocontrast agent, something that contains iodine. Iodine is a large atom. So if you have a lot of iodine in a solution, it will show up on an X-ray. They've injected that solution into the ureters that lead up to the kidney, and we're seeing abnormalities in the ductwork that collects urine within the kidney. Another example, here we have a patient that is being given a solution of barium to drink. Barium is quite dense, so we wait a while until that material makes its way to the large intestine, and it allows us to see the large intestine using X-rays. We wouldn't normally be able to see this level of definition. So the barium has coated in a thin film the sides of the large intestine, and we can see all the folds and so on. And we can look for any abnormalities. So this might help us identify a tumor, might also help identify if the intestine is twisted or if it's folded unusually. So if the intestine gets twisted somehow, that can cause a constriction, and that can block the intestine, which is pretty serious. So it's nice in that we can get a good image of the shape of the intestine without having to actually take out a scalpel and cut the patient open. And when it does come time for surgery, if that's required, we know exactly where to go. There are machines out there that can take multiple X-ray scans very, very quickly, thousands of scans, and then take all of those scans and use the information gathered by those scans to create a three-dimensional image. That's something known as a CAT scan or CT scan. And the image here, again, a pretty bad joke, but we've got a lion that's going into a CAT scan. This is quite literally a CAT scan. And down the bottom there, we're also going to talk about PET scans. Yeah, not quite what you're seeing there, but we'll get to that in just a second. Computerized tomography, that is a CT scan. Quite commonly known as a CAT scan. The A is just thrown in there because it sounds kind of nice, but the A doesn't actually stand for anything. So with a CT scan, what you're doing is you're taking thousands of X-ray pictures. Each X-ray picture is a two-dimensional image. It's basically a slice through the animal, the human, whatever it is that you're looking at. And then all of those slices are put together, they're laid over each other to create a three-dimensional image by a computer that's connected to this machine. So tomography is the construction of images using planar sections. And what I mean by planar is just they're in one plane. We're going to take a lot of sections by focusing in on one layer of the specimen, and then we're going to put all those planar sections together to create a three-dimensional image. Positron emission tomography, or PET scans, can also be used to generate two-dimensional planar sections. And those sections can be compiled together to produce a 3D image, just like we saw with CT scans. But PET scans are quite different from CT scans. CT scans are generated using X-rays. PET scans are generated using radioisotopes. And radioisotopes are forms of an atom that are unstable. They're radioactive and they give off high energy particles that we can detect. Now, the level of radiation that's used is fairly low, so it's not that dangerous. But what's generally done is that a patient is going to be given a radioactive metabolite. A metabolite is something that can be broken down by cells and used as a source of energy. The most readily available source of energy for cells is glucose. They can break it down very, very easily. So what you can do is take glucose, and the glucose is manufactured so that it contains a few radioactive isotopes. Cells that are very, very hungry for metabolites, cells that are very, very active, are going to gobble up that glucose, and that radioisotope within the glucose is going to accumulate within those cells. And now, when we look at the patient and we have sensors for the particles that are given off by that radioisotope, we'll see where those really, really active cells are. So as an example, let's say you've got a patient, you think they've got a tumor. Tumors consist of cells that are very, very rapidly dividing. That's essentially what a tumor is. So if you have a tumor, it means that you have a population of cells that have decided to ignore signals from the body that tell them not to divide, and those cells are dividing rapidly. Cell division requires a lot of energy, so those cells soak up a lot of this radioactive glucose. And then when you look at the patient using sensors that detect the radioactivity, you'll see where that glucose is concentrated. So on the left here, you're seeing a three-dimensional scan of a patient that has a rather large, actually several large tumors within their abdominal cavity. And you're also seeing at the bottom there, you're seeing a concentration of this radioisotope. That's actually in the urinary bladder. So some of that glucose is being extracted by the kidney and it's being stored as urine in the bladder. The diagram on the right shows you the brain of a person that's doing certain things. You're seeing sections through the brain, and you're seeing where certain activity is occurring. So if you look at thinking on the bottom there, you can see that we've got lots of activity shown as red in the front, in the frontal and prefrontal lobe, and that's where cognition, thought, occurs predominantly. So what they did here was they gave this person some of this radioactive glucose, and then they had them do different things. They had them look at pictures, they had them trying to remember things, they had them thinking about stuff, doing math problems, that kind of thing. And this allowed them to see what areas of the brain were very, very active during those tasks, so they could figure out what part of the brain was performing that function. Another really powerful means of looking inside a body without cutting into it is magnetic resonance imaging, MRI. This can give us really fantastic three-dimensional views of the internal anatomy. So that's a standard MRI machine there. That big donut contains some very powerful magnets that are actually cooled down with liquid hydrogen. This is an incredibly complicated machine. When electrons are moving, they generate magnetic fields. For instance, even when they're just moving around the nucleus of an atom, they generate magnetic fields. Magnetic resonance imaging uses radio waves to excite hydrogen atoms specifically, and it picks up on changes in the magnetic fields that are generated when these atoms are excited. What MRI does is it shows you where hydrogen atoms are concentrated, and that's going to be where water is concentrated, because of course water is 2 parts hydrogen, H2O. So when we're looking at an MRI, what it's showing you is the distribution of water. And in this image here, the red areas are areas that don't have a lot of water, and the blue areas are areas that have a lot of water. So you can see the skull is red because there's lots of minerals there. There's not as much room for water. The corpus callosum, which is a rather dense connecting structure between the 2 hemispheres of the brain, is red in the middle. But basically what this allows you to do is, again, to see three-dimensional structures within the body without opening up the body. Some advantages of this is that you don't have to use any radioisotopes like you would do in PET scans. PET scans, again, they're using small concentrations of radioisotopes, but those radioisotopes are radioactive. And if we look at X-rays, X-rays are ionizing radiation. They're high energy radiation. Again, in the amounts that they're used shouldn't be a problem, but if you have lots of X-rays done on you, that might be cause for concern. Okay, so that's a few ways of looking at the gross anatomy of a patient without actually cutting open and harming the patient. Let's look at histology next. Remember, histology is the study of tissues. So in this case, we do need a sample of tissue. But we can go in and we can take a little biopsy, a little chunk of tissue, a tiny little chunk of tissue. The patient won't miss it. And then we can take that and slice it up into thin little pieces and look at that under the microscope. And you're seeing an example of that right here. So this is a little section of the kidney, and we're seeing little tubules that are involved in collecting urine. We will spend a fair amount of time looking at histological sections in the lab. So you'll have a chance to look through microscopes and see sections of tissue yourself. Generally what's done is the tissue is taken, it's preserved, and then it's embedded in something, usually either paraffin wax or epoxy or plastic of some sort, and then very thin sections are cut, and then those sections can be looked at under a microscope. So a few examples here. In the upper left, we've got section through the brain. So you see lots of wrinkles and folds there. Those are the folds on the surface of the brain. This specifically is the cerebellum, the little bit at the back of your brain. In the upper right, we've got a section through the testes. And these cells that you see here in the middle of these tubes are actually developing sperm cells. On the bottom left, we've got something called areolar tissue. We've got these strands here that are actually strands of collagen. They're very, very long proteins that are twisted around each other to form cables. And then in between that, we've got cells that are known as fibroblasts. And those are cells that wander around in this loose connective tissue and produce those fibers. Don't worry too much about this stuff just yet. We'll come back to this, but you'll know what all of these are later in the course. And finally, in the bottom right here, we've got a section through the lining of the intestine. And the intestine has lots of folds in it. So those are known as villi. And those folds help increase the surface area. There's some great big white things here. These are actually individual cells that produce mucus. And the mucus gets dumped onto the surface, and it helps protect the inner surface of the intestine and keep things moving and flowing. If you study histology, you are a histologist. And what we're seeing here is the product of a histologist that has way too much time on their hands. They've managed to find some little cavities and so on in the slides they're looking at that look like letters, and they've made their own little histological alphabet here. Let's get a bit smaller and zoom in a bit closer. Cytology is the study of cells, also known as cell biology. If you're a cytologist, this is what you do. We've got a few examples at the bottom here. On the left, we've got a Purkinje neuron. This is one cell. It's a large cell with many, many projections that can connect to hundreds of thousands of other cells. This is something that's found in the brain. It's involved in cognition or thought. Next to that, we've got an assortment of blood cells here. So the smaller ones are red blood cells, and then we've got a few different types of white blood cells. Next to that, we've got a sperm cell fertilizing an egg. The egg cell is actually at least 100 times bigger than the sperm cell. So cytology, as I mentioned, this is cell biology, and we will spend some time on this. This is one of our topics. You'll get to know all the parts of the cell if you don't already. So this is a typical eukaryotic cell here. Eukaryotic cells have a nucleus. And we could get smaller than the cell. We could look at the molecules that make up the cell. Molecular biology is the study of how DNA works, how DNA is used to make proteins, and what those proteins do, basically. This is my area. I won't get to talk about it nearly as much as I'd like, but we will spend a bit of time on it. So we've got an example here on the left where we've got DNA that's being used to make messenger RNA, and the messenger RNA is going to be used by ribosomes to make protein. And that's kind of the summary of molecular biology. So molecular biologists study all the steps that go on in that process. Biochemistry, we're looking at things like amino acids and nucleotides and so on. Organic chemistry, kind of the same thing. We're looking at usually smaller bits, and we're just looking at the chemical reactions that occur between those bits. And chemistry, we're looking at smaller molecules as well. So we can look at large molecules in chemistry, but we're also talking about interactions between individual atoms. Let's continue with some more basic concepts. Remember that when we first started this topic, I was talking about the idea that anatomy and physiology are linked. Form meets function. I talked about how the lungs, instead of being just simple bags, contain lots of tiny little sacs called alveoli, and that greatly increases the surface area of the interior of the lungs. It increases the area over which gases, carbon dioxide and oxygen primarily, can be exchanged between the atmosphere and the blood. And this surface area to volume ratio is really, really important in a number of processes that go on within the body. So let's look at a single cell. So in part A, we have an amoeba here. And an amoeba is a free-living cell. It's an entire organism. And it has to exchange materials with its environment. It has to exchange water. It has to move oxygen into the cell. It has to get rid of carbon dioxide. It has to take in nourishment. It has to get rid of waste, etc. We have this volume, this fixed volume of the cell that needs to exchange materials with its surroundings. The greater the surface area, the faster it can do that. So think of it as, every square millimeter of surface area can exchange X amount of material every second. More surface area, a greater rate of exchange. Now let's look at a multicellular animal. So an animal that consists of more than one cell. And the example you're seeing here on the right is a very simple animal. It's something called a hydra. It's very similar to sea anemones. It's in the same group. It's something that we have around here. Actually, you'll find it in ponds and rivers. It's fresh water. It's only about a centimeter or so in size. And it consists of two cell layers. We have an outer layer, the epidermis. We have an inner layer that's making contact with this central body cavity where it absorbs nutrients. But the thing is, now we have, instead of one cell, we have lots of cells. It's a little more difficult to exchange material because not all of those cells are exposed to the external environment. So in the epidermis, you can see we've got cells that are roughly square shaped. One side is exposed to the external environment, but the other three sides are not. And that's going to limit the rate of exchange. But what could happen in a lot of animals, and does happen as we'll see, is that the cells will grow extensions. So the cells inside your intestines, for instance, have little appendages, little extensions called villi, which will increase the surface area of the cells and allow exchange to occur more rapidly. Let's take a look at a very simple mathematical example. So in this table here, in the left-hand column, we have a cube that is 1 unit by 1 unit by 1 unit. So just for the sake of argument, let's say it's 1 centimeter by 1 centimeter by 1 centimeter. So picture that cube. How would you calculate its surface area? Well, each one of the sides is going to be 1 centimeter by 1 centimeter. So 1 times 1, that's 1 centimeter squared. There's 6 sides on a cube. Add them up, we have a surface area of 6 square centimeters. The volume is going to be 1 by 1 by 1. So 1 times 1 times 1, that's 1. So it's got a volume of 1 centimeter cubed. The surface to volume ratio is going to be 6. So we have 6 centimeters squared divided by 1 centimeter cubed equals 6. Now what if we increase the volume and increase the surface area by looking at a bigger cube? So now we have a cube that's 5 by 5 by 5. The surface area of each one of the sides is going to be 5 by 5, that's 25. Multiply that by 6, we get 150 for our surface area, so 150 centimeters squared. The total volume is going to be 5 by 5 by 5. That gives us a value of 125 centimeters cubed. Now, if we figure out the surface to volume ratio now, we get a value of 1.2. It's much lower than what we had for the smaller cube. That is going to affect the rate at which materials can be exchanged with the outside environment if we were talking about a cell. So if we were talking about a cell, we're going to have a bigger cell now, and we have more material on the inside. We have more going on the inside, more metabolism. We have a greater need to exchange materials, but we don't have a proportionally larger surface area. We have this smaller ratio between volume and surface area. And the cell is going to struggle to exchange materials with its environment. And this limits the size of cells. Cells can only become a certain size typically without either having to reduce their metabolic rate or divide. If a cell divides, now we have that same volume divided into 2 cells, and we've increased the surface area to volume ratio. So what multicellular organisms have done, so we are multicellular organisms of course, is they have evolved to consist of lots of small cells. An amoeba cell is much bigger than your typical human cell, and the reason for that is this surface area to volume ratio. So if we took that same example where we have 5 units in each direction, so we have a cube, we have a block of material that is 5 by 5 by 5, and instead of making that material out of one cell, we make it out of a whole lot of cells, We divide that up into 1 by 1 cells, and then we allow fluid to seep between those cells. We have brought our surface area to volume ratio back to the number 6. And these cells can exchange materials with their environment much more readily. That's what happens in your body. You have lots of little cells instead of, a smaller number of big cells. You have the movement of fluid between the cells so that every one of those cells can exchange materials with its environment. You are essentially a colony of cells. You consist of more than 100 trillion cells, and all of those cells have the same requirements. They need to take in metabolites, they need to get rid of waste, they need to take in oxygen, they need to get rid of carbon dioxide, etc. So your body has to maintain an internal environment and make sure that every single cell in your body has access to what it requires. And that's done by 4 systems, fundamentally. There's other things involved, but we're just going to look at the 4 main systems just really briefly. So what you're seeing here is a very generalized or idealistic version of an animal. An invertebrate, a vertebrate, whatever. We all have basically the same needs and requirements. First of all, there has to be a digestive system of some sort, or there has to be some way to absorb nutrients. So we'll talk about this later, but basically the digestive system is just a tube that passes through your body. It's a tube inside a tube. It's a tube that's surrounded by a fleshy tube, which is you. You have one end of the tube, which is the mouth, the other end, that is the anus. Food comes in, nutrients are broken down and absorbed, and anything that can't be absorbed or any additional waste that you might want to get rid of leave through the anus. Those nutrients are dumped into the circulatory system. Then we have a respiratory system. So that could be gills if we're talking about a fish, for instance, or it could be lungs if we're talking about a mammal like ourselves. And oxygen comes in. Oxygen is going to pass into the blood of the circulatory system. Carbon dioxide is going to be removed. And then we have an excretory system, and what I'm talking about here is the urinary system. So we have kidneys that have many, many fine blood vessels within them that are going to exchange materials with the circulatory system again. Wastes are going to be dumped out of the blood into the urinary system, and also the urinary system can control what leaves the body. It's going to retain things that the body needs. Notice that the 3 systems I just talked about, they're all linked together by the circulatory system. So the circulatory system is going to pick up oxygen, it's going to pick up nutrients, it's going to carry that to every single cell. It's going to pick up waste from every cell and carry that to the kidney. It's going to pick up carbon dioxide from every cell and carry that back to the respiratory system to be released and disposed of. So we have these 4 fundamental systems, 3 systems that are intimately linked by the circulatory system. As you've no doubt heard before, your body is mostly water, and that's because your cells are mostly water. And your cells are surrounded by fluid as well, known as interstitial fluid. That's the fluid that bathes the cells, and that fluid is produced by blood vessels, by very small blood vessels known as capillaries. So the liquid portion, the fluid portion of your blood is known as plasma, and that seeps out and gives rise to the fluid known as interstitial fluid. The cells themselves, mostly water as I've mentioned, and some protein, most notably enzymes. So the enzymes are responsible for regulating metabolic reactions. Now there's other stuff as well, DNA and RNA and so on, and we'll get to all that. But it's kind of useful for the moment to think of your cells as just kind of little water balloons surrounded by water. Homeostasis means to maintain an equilibrium, maintain an internal environment. So your cells need to maintain their internal environment, and also the body as a whole needs to maintain an internal environment. So for instance, temperature. We're mammals, and like other mammals, our body temperature stays pretty close to 37 degrees Celsius. It may go up a little bit, a half degree, it may go down a half degree, but if it gets way out of whack, way out of line, that's going to cause problems. And that's because the chemical reactions that occur in your cells work best at a temperature of 37 degrees. The enzymes responsible prefer that temperature. Your body also has to maintain a certain salinity and a certain pH in the interstitial fluid and in the blood. We'll talk more about homeostasis as we go on in the course. In fact, it's a major theme. We'll talk about the different systems that are found in the body and how they help maintain homeostasis throughout your body. Homeostasis is maintained by feedback systems. What I mean by feedback is, let's say there's a change that disrupts the normal balance. There's going to be a response that brings it back towards normalcy. That is feedback. So imagine that, let's say we're talking about the stomach. The pH of the stomach is very low. It's quite acidic. It's more acidic than any other part of your body, and that's because acidic conditions help with the unraveling and digestion of proteins. But the stomach normally has a pH of 2 or even lower. Now imagine that for some reason the pH gets out of whack. Maybe you've eaten something that's very basic and the pH gets up to 3. Well, what's going to happen is there's going to be a signal sent to your nervous system, and that signal is going to induce cells within the lining of your stomach to release more hydrogen protons, to manufacture more hydrochloric acid, and that's going to bring the acidity back down. And then once the acidity is back where it should be, your body will stop responding so that it doesn't keep lowering it. So in general, we have a stimulus that's going to be received by a receptor. So it could be a chemoreceptor in the example I just used that senses pH. There's going to be a signal sent to the control center, which could be the nervous system. It's not always, if we're talking about a single cell, for instance. And that's going to bring about a change. So it's going to trigger a gland, for instance, to produce more hydrogen protons or to produce more hydrochloric acid. And that gland, the thing that's being affected to bring about a change back to normalcy, is known as an effector. And we'll get back into this in more detail later. So looking at the flow chart on the right, we have some sort of stimulus that's going to disrupt homeostasis. So we're not at the norm anymore. We have receptors that are going to receive that input. They're going to tell the body that things have changed, or tell the cell that things have changed. So remember, homeostasis is maintained within individual cells, but also within the entire body. We have a control center that receives that input and is going to bring about an effect. So there's an output that affects effectors, and the effectors are going to do something that brings us back to the normal situation. Now, the one thing to note is this doesn't happen instantly. So there is going to be a bit of a fluctuation, but the fluctuation in pH or temperature or whatever we're talking about should be fairly close to what is normal. So temperature, for instance, 37 degrees Celsius is the normal temperature for a human being. But if it gets up to 37.5, even 38, that's not out of the ordinary. That's not something to be concerned about. If it drops down to 36, that's not necessarily something to be concerned about. But if temperature goes up to 38, your body will be bringing it back down. So if you measure your temperature over the course of the day, you'll find that it fluctuates up and down a bit, but it doesn't get very far from 37 degrees. Negative feedback is the type of feedback that reverses a change, and that's what I've been talking about so far. That's what is responsible for homeostasis. A nice example would be the thermostat in your house. The thermostat is set to a certain temperature, and let's imagine that you set that to 20 degrees Celsius. If the temperature drops, so let's say it's winter, it's quite cold outside, the temperature drops down to 19 degrees. What's going to happen is your radiators or your furnace are going to come on, and that's going to increase the temperature in the house. When it gets back up to our set point, which is 20 degrees Celsius, the radiators or furnace should shut off. And that'll keep happening. The house will keep cooling, and this will keep happening, and the temperature will stay at 20 degrees. But it won't stay at exactly 20 degrees, as you know. Depends where your thermostat is, for one thing. So parts of your house may be colder or warmer, but also the system takes a little while to respond. So perhaps the thermostat might not kick in until it gets down to 18 degrees. And maybe it's not going to shut off until the average temperature of the house is 22 degrees. This is a dynamic system. So we have the system cooling constantly, and then we have a heating system kicking in. It's never going to be exactly what you want. It's never going to be exactly 20 degrees, but the temperature should hover around 20 degrees. It will be within certain limits. And how good it is depends on how good your thermostat is, and also how much airflow you have in your house. But this is the same thing that happens in the human body. Your temperature goes up and down. It's never exactly 37 degrees, but it's close to 37 degrees. Let's talk about the same process in your body. You have a thermostat in your body. The hypothalamus is part of your brain, and it monitors blood temperature, and it's responsible for thermoregulation. So thermo meaning temperature, the regulation of temperature, just like the thermostat in your house. So let's imagine that your body gets too warm. You're exercising, you're out in the sun, whatever. If your body gets too warm, there are sensors in the hypothalamus that are monitoring the blood flow that flows through that structure. And once they sense elevated temperature, they activate cooling mechanisms. So they send out a signal to the rest of your body, and they tell your sweat glands to start producing sweat, and the sweat is going to be dumped onto the surface of your skin. It evaporates. As it evaporates, it steals heat away from your skin. The hypothalamus is also going to tell blood vessels to dilate, to open up next to the skin and allow more blood flow into the skin. So you may become kind of red and flush. The purpose of that is so that blood can release heat into the atmosphere, release heat through the skin. And that's going to bring the temperature back down. The hypothalamus will sense that the temperature has dropped, and it will stop doing those things. Now let's say the opposite occurs. We have decreased blood pressure because maybe you're out on a cold day. What the thermostat in the hypothalamus is going to do is it's going to, first of all, cause blood vessels near the skin to constrict. So blood will be kept away from the skin, and your skin will be kind of pale as a result, and at a risk of frostbite too, but we'll talk about that later. But this is going to reduce the exposure of your warm blood to a cold environment. Also, the hypothalamus is going to tell your muscles to contract, to twitch, and that creates shivering. And what that does is it creates heat. So your muscles will start twitching, and as they do that, they generate a lot of excess heat, and that will help heat the body. And once again, once the temperature goes back to normal, the hypothalamus should shut off. But none of these processes are instantaneous. There's a bit of a lag, and what that means is that the temperature of your body is never exactly 37. It's between 37 and 38. And once again, we call that a dynamic equilibrium. Dynamic means movement. Equilibrium means the same. But we have a bit of shift here. So we have movement of the equilibrium, very slight movements, but it averages out to 37 degrees. Negative feedback is where we have a self-correcting system. So we have a change, and that brings about a negative change that brings us back to where we started. Positive feedback is where we have a stimulus. That's the result of a change. The response is to heighten that change. It's a positive response. So it reinforces the change. This does not result in equilibrium. And as you might imagine, positive feedback loops are not that common in living things. And the reason for that is because living things typically want to maintain an internal environment. They want homeostasis. But sometimes there's special events that warrant a quick and rapid change to bring about something new. An example here in this diagram, we've got a positive feedback loop that occurs in the production of milk in a lactating mother. So we've got a stimulus, and that would be suckling by the baby. And sucking at the nipple is going to stimulate mechanical receptors, we'll talk about them later, within the nipple. These receptors are going to be stretched. They send off a signal, and they are going to excite the hypothalamus into bringing about the release of oxytocin, which is a hormone. Oxytocin is dumped into the blood, and it will target cells within the breast and tell them to basically release more milk. We have milk released. That's going to cause more suckling by the baby. Again, we're going to have these touch receptors, these mechanical receptors being activated, sending more signals to the hypothalamus. So we can go from a point where we start with a rather small stimulus from the baby, and within just less than a minute, we have lots of milk being produced because of this positive feedback loop. When the baby stops suckling, this will eventually return to normal, although that doesn't happen immediately. Another good example, and it's very similar actually, would be labor. So when a baby is about to be delivered, what happens is typically, if the baby is oriented correctly, the baby's head will push on the cervix. There are stretch receptors in the cervix and the uterus, and those stretch receptors will send a signal again to the hypothalamus telling it to release again oxytocin, and the oxytocin will bring about a dilation of the cervix. The cervix will widen, and this is a positive feedback loop that brings about a change. It brings about the delivery of the baby, and in this case that's an irreversible change. The baby's not going back in. So positive feedback brings about a change, and it brings about a change fairly quickly, and in some cases that change is irreversible. So finally, let's take a look at some very important anatomical terminology. We're looking at directional terminology here. These are terms that you'll be using throughout the course, so make sure you understand these, and these are terms that you'll use throughout your career. First of all, let's talk about planes of section, or axes. What we're talking about here are two-dimensional planes that we can use to divide a body, or divide an organ, or to divide a cell. If we're looking at a human body like we have here, we have the long axis, which would be the longest dimension of the body. So if you're standing upright, that would be the height of your body. If we cut perpendicular to that long axis, that would be a transverse plane of section. If we cut through that longitudinal axis, dividing something into essentially a front and a back, and I'll define that a bit better in a moment, then we're looking at the frontal axis. And if we divide through along that longitudinal axis, and we divide the body into two equal halves, let's say, that's a sagittal plane of section. Now, if we're just slightly off center and we divide the body into two unequal halves, then that's a parasagittal section. But I'll come back to that in just a second. But realize that we have three planes of section because we have, of course, three dimensions. We're three-dimensional objects. So we have transverse, we have frontal, and we have sagittal. Again, the body can be divided into these three planes because we're three-dimensional objects. We have the frontal, sagittal, and transverse. Now, as is often the case in biology and pretty much always the case in medicine, these terms have other names. Frontal can also be referred to as coronal. Corona means crown. Think of the beer. It has a little crown on the bottle. Think of coronavirus. The coronavirus has a crown of proteins that stick out from its envelope. We'll talk about that more in 105 if you're doing 105 with me. At the top here, I have a little skull and I've highlighted the coronal suture. This is where the frontal bones and the parietal bones meet. And if you notice, this is pretty much where a tiara would sit. If you were wearing a tiara, it would sit right on top of that suture, which is how it got its name. It's also a good example of a frontal section. If I sectioned a skull right down here, that would be a frontal section. Now, a sagittal section, also called a longitudinal section, is going to be perpendicular to that coronal section. A transverse section is going to be perpendicular to both the sagittal section and the coronal section. Now, these terms can be applied to anything that has a longitudinal axis. And what I mean by that is anything that has at least one dimension that's longer than the other two. You couldn't really apply these terms to a basketball, for instance, but you could, if you really wanted to. I don't know why you'd want to, but if you wanted to, you could apply this to a hot dog, for instance. So with the hot dog, the longitudinal axis is going to be through the middle of the wiener. The transverse or cross-sectional axis is going to be perpendicular to that, so it's going to be across here. So that would be our transverse. Our longitudinal or sagittal would be along here. And then I'm not sure how well I can draw this. Hopefully you understand what I mean here. Our frontal section would be through the bun and the wiener across here. So let me try to draw that as a square. I'm not sure how well that worked. Hopefully you figured out what I meant there. It'd be easier to do with an actual hot dog in front of me. So here we have an unfortunate individual that's being sectioned mid-sagittally, a cadaver, fortunately. So we've got the head and shoulders of a cadaver, and it's being sectioned right down the middle through the nose along the mid-sagittal plane. And in humans and other bilaterally symmetrical animals, the mid-sagittal plane is the only plane of section that will give us two equal halves. Here are some more cadavers. So at the left there, we've got a transverse or cross section. So remember, a transverse section, also called a cross section, is perpendicular to the longitudinal axis. You can see the section here is through the brain and the brainstem and the eyeballs. On the right, we've got a frontal section, frontal or coronal section. So this is running along the longitudinal length of the human, but it's perpendicular to a sagittal section. So let's take a look at these sections together. And let's just label these here, I guess. We've got number 1 here. Number 1, what we're doing is we're sectioning the brain in this plane. So you're seeing the brain at the top there from a few different angles. So we've sectioned it like that. That would be a frontal section. The next here, let's look at the upper right. So I'll call this 2. Here, we're sectioning the brain like this, along here, or along here if you want. That would be a transverse section. And then finally, for the last diagram here, diagram 3 we'll call it, we're sectioning right down here. So we're sectioning down this plane, or in this diagram, it would be down here, and that would be a sagittal section. And because we're right in the middle, that would be a mid-sagittal section. If for some reason, and you wouldn't normally do this, but if for some reason we decided to do a section like that, that would be called a parasagittal section. Para means beside. Think of parallel. So it's not in the middle, but it is sagittal. Now I'm not sure where this is from. I have seen this done a few times. I've witnessed this in a few different museums that I've visited, but I didn't actually take this picture. But this is an individual that has been sectioned in the frontal or coronal plane. So we've got several thick sections of one preserved cadaver here. Now that we've taken a look at the planes of the body, let's take a look at some directional terminology. Directional terminology is used to compare the location of 2 points. And it's kind of similar to what you use in real life. You can say that I am east of you, or my house is west of your house. Or you could say that I'm above you, or I'm below you, or I'm behind you, or I'm in front of you. It's the same kind of thing, but it's with reference to the body in this case. So we can say that something is superior versus inferior. Now imagine a person that is standing upright. We could say that their head is superior to their knees. Or we could say that their feet are inferior to their knees. So inferior is below, superior is above. There are other ways of saying this. So cranial is Latin for having to do with the head. Cephalon is Greek for the head as well. So we could say that your heart is cranial to your hips, or your heart is cephalic to your hips, which wouldn't be said very often. But cranial and cephalic can be used in place of superior. It means towards the head, basically. Likewise, caudal means towards the tail. Now, humans don't have a tail, technically, but we have a coccyx, which is a remnant of the tail. Things are a bit confusing, though, if you use caudal with humans, because we stand upright. We are bipedal, which is very different from other organisms. So if I was talking about a cat, and I said that the ribs are caudal to the jaw, that would make sense. But using caudal in humans sometimes can be a bit confusing, so it's not usually done. Next we have anterior versus posterior. Anterior is towards the front. Again, picture someone who's standing upright. Posterior is towards the back. So I could say, perhaps, that your chin is anterior to your ears, or your ears are posterior to your chin. Then we have superficial versus deep. Now, this is a kind of an odd one because we're talking about location as it refers to the surface of your body. So we could talk about your heart, which of course is underneath your breastbone. So your heart is deep to your breastbone. Your breastbone is superficial to your heart. Superficial means close to the surface, deep means below the surface. But again, these are directional terms. When you talk about a superficial cut, the meaning of that's a bit different. It means it's just on the surface of the skin, but again, we're using these terms as directional terms referring to locations. Medial versus lateral. Medial is towards the midline. So think of the mid-sagittal plane of section. If something is close to that, it's medial compared to something that is far away from the midline. So let's come up with another example here. I would say that your heart is medial to your shoulders. Your shoulders are lateral to your heart. Proximal versus distal. Now this is an interesting one. And this relates normally to limbs, but it can relate to anything that's attached at one end and free at the other end. Proximal means towards the point of attachment. Distal means away from the point of attachment. So let's look at your arms. Your arms are attached to your body, but they're free at the end. Your fingertips are free to wiggle around. Your fingers are distal to your shoulder. They're away from the point of attachment. your, let's say your elbow is proximal to your fingers. That means that it's closer to the point of attachment. I'll use one more example here. Your wrist is distal to your elbow, but your wrist is proximal to your fingertips. So here's a nice little example of how these terms might be used. On the left, we have a person that's facing us, and we have a line right down the middle, which would be a mid-sagittal plane. Medial is towards that mid-sagittal plane, lateral is away from it. Now, when you're discussing anatomy of a person, you also want to include right and left if that's appropriate. So we could talk about veins as being either right or left. We talk about limbs as being either right or left. When you do that, make sure that you remember it's not your right or left, it's the person's right and left that we're referring to. With limbs, we can use the terms proximal and distal to refer to where points are located in relation to the point of attachment. Now, we could also use this with respect to some organs as well. I'll show examples of this later, but the pancreas, for instance, it's attached at one end and free at the other end. We could use proximal and distal in relation to that. Also, proximal and distal are used with reference to the digestive system sometimes. The point of attachment, and it's not really a point of attachment, but is considered to be the mouth, and then we have a point of exit, which is the anus. So something is proximal if it's located closer to the mouth, something is distal if it's located closer to the anus. So the stomach would be proximal to the intestine, that sort of thing. That's not used as often. I should also mention that you can use proximal and distal for the penis as well. It's considered an appendage. We also have a diagram here that's showing a woman from the side, and you can see anterior is towards the front, towards the belly. It's also called ventral as well. Dorsal is towards the back. And ventrum and dorsum literally mean stomach and back. You may be dissecting quadrupeds, four-legged critters, in the lab. So I just wanted to point out that the directional terminology is a little bit different in the two, and that can get kind of confusing. Quadrupeds are animals that walk on four legs, the majority of animals. Bipeds are animals that walk upright on two legs. So us, for instance. There's not too many other examples of bipeds. I mean, I can think of kangaroos and T-Rexes. Although technically, I guess kangaroos are tripeds because they use their tail as well, but never mind. So with quadrupeds, we tend to use the term cranial and caudal, and we use the term dorsal and ventral. We don't use those terms as commonly with humans, but I just put this here just in case you run across it and you get a bit confused. Now, it's important to note that when we're referring to the human body and using these anatomical terms, we always assume that the body is in the anatomical position, regardless of what position the body is actually in. I mean, if you have a patient lying face down on a bed, doesn't matter, you're referring to the direction or the location of things based on the anatomical position. So the anatomical position is someone standing upright, standing flat on their feet with their palms facing forwards, which is important. That's rather important when it comes to looking at the hands, right? If their palms were faced backwards, then the position of the thumb relative to the other fingers would change. So we always have the palms facing forward, the thumbs out. So let's use a few more examples here. So let's look at the elbow. The elbow would be lateral to the stomach. The elbow would be lateral to the ribs. Looking at the arm, the humerus would be proximal to the ulna. The metacarpals would be distal to the radius. What else do we have here? Let's look at the lung. The lung would be superior to the gallbladder. The rib cage would be superior to the urinary bladder. The colon would be inferior to the heart. And I invite you to do this kind of thing, look at your own anatomy, maybe get a friend and, point at different parts of their body and describe how they relate to other parts of their body. That might be, I don't know, a fun Saturday night. I'm not sure. But whatever helps you learn this stuff. I should mention that there's one organ that's kind of weird when it comes to directional terminology, and that's the penis. And I just mentioned this because you're unlikely to see it anywhere. I'm not sure why it's not mentioned in textbooks and other places. The penis in the anatomical position is erect, and I mean very erect. It should be pointing right up so that it's pressed against the abdomen, because the surface of the penis that would make contact with the abdomen during a rather extreme erection would be the dorsal surface, and the bottom surface would be the ventral surface. Just a weird little aside. Very important though, make sure you know the anatomical position. The anatomical position is someone standing up, hands at the sides, palms facing forward, feet together. And the one error with this particular figure is that guy should have an erection. Also though, if someone is in the anatomical position and they're lying face up, we call that someone in the supine position. And if they're lying face down in the anatomical position, we refer to that as the prone position. And we'll come back to this. This is important when we're talking about movements of the forearm and hand. Does anyone remember planking? Maybe it was before your time, or maybe you're just fortunate enough to have forgotten all about it. But planking was an odd trend where people would lie pretty much in the prone position in very awkward places. I say it's almost the prone position because, of course, if you were literally in the prone position, your palms would be faced down towards the ground.
Oh, for God's sake. Planking is a very stupid and dangerous trend. Basically, you lie like a plank in weird places. That's it. Sometimes you get run over. Welcome to the internet.
Smart, right?
Very smart.
This has got to stop.
I can't get down.
No one should be planking at all.
Thank you. Yes. Dwight, my enforcer, my number 2, can you take care of this?
Say no more.
Kids, don't try planking. It's dangerous.
Especially with me around.
And who doesn't love The Office? Lots of people probably. Anyway, that's it for me. At the end of all my PowerPoints, I will have a terminology list, and that's what you're seeing right here. These are the terms you should know. So as you go through the study guides that I'm going to provide, and as you go through your textbook and so on, reading up to get additional background info, these are the terms you should focus on. If you come across terms that are in your textbook that aren't on these terminology lists, they're not something I'm going to test you on. When I make my tests, I have terminology lists in front of me, and I draw from that to write my multiple choice, write my matching, write all the questions that I use. So this is your guide for what you should be looking at. And I just want to say this isn't an easy course. I would say typically 1/3 of the people in this course won't pass it. That tends to be what I see year after year. And I'm really hoping that's not you. Stick with it, review often. You need to see the material several times. So read your textbook ahead of time, watch the PowerPoints, review the PowerPoints, use them to make notes, etc. And just stick with it. You don't want to lag behind. So you need to study and review often. And of course, I am here to help you whenever and however I can. Okay, we'll see you in the next lecture.